Hello, and welcome to Chapter 4, Part 2, where we're going to look at some different laboratory techniques that we use to study the growth of bacteria. In order to research bacteria, the first thing that we have to do in the laboratory is we have to isolate that bacteria from other organisms that may be in the sample. So this is done with a technique known as a streak plate isolation, sometimes referred to as an isolation streak plate. In the laboratory, when we grow bacteria, we use either solid media, which is agar, kind of this really hard jello. Agar is extracted from seaweed and seagrasses out in the ocean. It's also used in cooking as a solidifying agent. But this agar is supplemented with nutrients and elements that are needed for bacterial growth. We can make all kinds of different types of agar depending on the microorganism. We also have broths. Broths are a liquid form of media that's just water with supplements in it as well. Of course, we always need to use aseptic technique in the laboratory. We'll learn more about that in our actual micro labs themselves. So when we grow bacteria in the laboratory, this brings us back to that idea of a growth curve. And in a growth curve, there are actually 5 phases of growth. And this is what's occurring in a closed environment. Remember, we're looking at bacteria that's being grown in the laboratory. So imagine a test tube with some liquid in it. And inside of that liquid, we put a couple of bacterial cells. So in that test tube, we're not adding any new media. We're not taking any media out. We're just simply putting the bacteria in there, putting them in the proper temperature, and allowing them to grow. And when we do that, we get what's up here is kind of what we call a standard growth curve. So we have our y-axis here, which is a logarithmic scale for number of cells. So this is that log 10 value, right, of our bacteria. And on our x-axis down here, we have time. Right? So we're looking at growth, number of cells over time. If we graph these numbers of cells, if we went after so many hours, we would take samples and get an idea of the growth of bacteria in there and convert those into log values, we would see a typical curve like this. And this is our five phases. The first is the lag phase. In the lag phase, there's no growth occurring. This is like the bacteria are moving in. And so right now during the lag phase, they're figuring out what nutrients are available. Do I need to break down proteins? Do I have sugars available? Do I need to ferment? Can I use cellular respiration? They're just basically getting a lay of the land. They're kind of feeling what's going on inside and what they have available to them. Once the bacteria have kind of settled in, so to speak, that usually only takes a few hours, they kind of settle in, they are able to begin producing the enzymes and other proteins necessary for metabolizing the nutrients that are present. So now the bacteria begin dividing. And in this sense here, during what we call the log phase or exponential growth phase, our bacteria are extremely metabolically active. There's a lot of metabolism going on because they need a lot of energy. There's also a lot of cell division occurring. So the number of cells within the population of bacteria in that test tube is increasing exponentially. So it's that log phase of growth. But eventually, some of those cells that divided a long time ago are going to start getting old. And in addition to that, we're going to start seeing an increase, because of that increased metabolism, we get an increased concentration of waste. So most bacteria will use fermentation, and when bacteria ferment, they give off acids. So what's going to happen is acids are going to start building up inside of that test tube. Now, many times we will add a buffer to the tube in order to prolong the log phase, because we might want to grow as much bacteria as possible. But with no buffer, we're going to start seeing the pH start to decrease in that tube, and that's because the bacteria are producing acids. Now, eventually, that acid waste is going to kind of hit a threshold. It's going to hit a level where it becomes, it can start to inhibit growth. We also have old cells. So what happens for a very short period of time, usually a very brief period of time, we reach what's called the stationary phase. And in this stationary phase, the number of cells that are resulting from division equals the number of cells that are dying. So for every cell division that occurs, 2 cells die. And we end up with a population that remains stable. So it's a stationary population. Death equals fission equals binary fission, and so we have an equal rate of both death and growth, and so our population remains stagnant or stationary. This is a very short period of time because during this metabolism is still occurring. So what's happening? We're going to start still see increases in that acid waste till it reaches this threshold that bacteria just cannot handle it. So in that case, they'll hit this death phase. And in the death phase, we'll see a rapid decline in population numbers. And then eventually we get what's called phase of prolonged decline, where our death rate starts to kind of slow down. So the death phase is that drastic drop, and then the prolonged decline is that more gradual drop in population numbers. And this is due to now we're not producing as much acid waste. A bunch of the cells that died are going to give off new nutrients. So there's now nutrients being released back into the environment for recycling. And bacteria are going to be able to, some organisms in that test tube are going to be able to sustain some growth. So we'll have the death rate kind of just decreases a little. Now what I wanted to show you is over here on the left, this graph I was talking about, this is the graph that's in your textbook. But down here below it, I have some pictures that are from some actual real data where they were looking at some different compound heavy metal effects on growth of bacteria. But there were a few things that were really kind of interesting in this research paper that I wanted to show you, and that was really about pH right here and dissolved oxygen. So in pH in particular, remember I was talking about how acid builds up because of fermentation. And what we see in pH, here they took the actual pH of the culture, and here it is over time. So at 0 to around here, right, I'm in my lag phase. My pH is neutral. It's around 7. But as I go up in my log phase, right, in my log, what happens to the pH? It starts to drastically drop. I get to kind of this stationary phase here where I start to see less of a drop in my pH, right? So this is stationary. And then I have a drastic decline. So this would be my death phase. And what do I have? Now my pH spikes back up. And then from there, I'd have my prolonged decline. And that, of course, corresponds with a slight increase in the pH back up. So even though from 7 to 6.2 might not be or down here to 6.0, but that's a 10-fold drop in pH. That's a pretty drastic decline in pH in a short period of time. So in pH, because acidic conditions drop the pH, we see that our culture here is we can follow our standard growth curve. It's just inverted. If I flipped this upside down, it would be a standard growth curve. Now in dissolved oxygen, if my bacteria are using, right, so we're looking at oxygen saturation, if my bacteria are using cellular respiration, I have my lag phase right here, and then I go directly into my log phase. Oxygen does not get replenished. So as oxygen drops drastically, it then will not come back up. Right? So we have this rapid, drastic drop in oxygen. So really the pH here was kind of cool because it really mimics that standard growth curve. So you do want to know the 5 phases of the growth curve, and you want to be able to recognize those on a graph of growth. Now, bacteria are also influenced by the environment. So an environmental influence on bacteria, one of the primary influences is going to be temperature. In temperature, you'll notice throughout this course that microbiologists like to classify bacteria a bazillion different ways. And so when we classify them based on what's known as their optimum growth temperature, right, optimum growth temperature, if we classify them by their optimum growth temperature, we come up with these 5 different classes. So let's start from the coldest to the hottest. So the first are the psychrophiles. And psychrophiles down here on our graph are in blue. And this graph is indicating rate of growth. So as we move across our x-axis, as the temperature increases, we see what happens to our rate of growth. And psychrophiles, they like it pretty cold. They can grow in about a -5 degrees Celsius temperature, so below freezing. When they get up here into the maybe 5 to 10 range, this is what, about freezer or super cold refrigerator temperature. And this is their optimum growth temperature. They actually like to grow in a very cold environment. So this is where you would see bacteria in the Arctic or Antarctic, in the ocean, in the northern hemisphere, where it's, or the North Pole area, that kind of thing, where it's really cold. Those bacteria can grow in that extremely cold environment. Once we get above around 5 or 7 degrees Celsius, the rate of growth is going to drop drastically. They can still grow up here at room temperature, but their rate of growth is going to be really very, very slow. Next are the psychrotrophs. The psychrotrophs overlap the psychrophiles in their growth, and they have a much broader range. They have a very wide range of growth. These are the organisms that grow well at room temperature, right? So up here, this is their optimum growth temperature up here. So if I bring this down, that's around 25 to 27 degrees Celsius, which is pretty average room temperature. So they like to grow at room temperature. These are the organisms that are in like your milk, that if you leave your milk out at room temperature, all of a sudden in a few hours your milk goes bad, whereas it can stay for days inside the refrigerator. So these organisms are more room temperature and refrigerator organisms that do things like spoil food. Now over here we have the really important ones, which are the mesophiles. And here we have our mesophiles' optimum growth temperature up here at the top. And if we go all the way down to our temperature here, we're looking at about 37 degrees Celsius, which is human body temperature. And that's really the body temperature of most warm-blooded mammals. The most warm-blooded mammals are in the 35 to 40 degree range. So this 37 degrees Celsius here is this optimum growth temperature. So mesophiles are where we see most of our pathogens. So most pathogenic bacteria are going to be mesophilic. Now notice these organisms can grow at room temperature. They can grow at lower temperatures, but their rate of growth is much slower. If we want to give them the optimum growth temperature, the perfect temperature to grow at, we would give them 37 degrees Celsius. And then of course we have the same process here in the thermophiles and the hyperthermophiles. These are extreme bacteria. They're found near like lava vents in the oceans, in those areas. They grow at extremely high temperatures. Now another environmental factor that's really important are the oxygenic requirements. Now bacteria can be aerobic, meaning they require oxygen. Anaerobic, meaning they do not require oxygen. They actually can't be near oxygen. Sometimes organisms can be both anaerobic or aerobic, and sometimes they're kind of neither. They're like in between. So let's take a look at how we would determine this. For determining oxygenic requirements, we use a test called an aerotolerance test. I'll just write it up here. And in the aerotolerance test, we use a large test tube. Right? So here I have a large test tube. And in that test tube, I fill it about 3/4 of the way with agar. Now, agar, when we make it, is in a molten form. Think of hot Jello. So it's really kind of viscous. It's in this liquid form. We pour it into these test tubes. We autoclave it to get all of the oxygen out of the media and to, of course, sterilize it. When we take it out of the autoclave, we put it in a hot water bath and we keep it hot so that the agar will remain in its liquid form, somewhere around 45, 50 degrees Celsius. So we keep the we keep the agar in this liquid form. Now, as this agar sits in the hot water bath, this is in liquid form here, it is exposed to the environment, meaning it's exposed to oxygen. So oxygen will begin to diffuse down into the media, and this creates an oxygen gradient. And so at the top of my tube and on the surface, I have a very high oxygen concentration, but at the bottom of my tube, I have no oxygen. So as I move down towards the bottom of the tube, the concentration of oxygen will decrease. I then take bacteria that's growing in a liquid culture, and I inject some of that bacteria into this liquid agar. And I kind of spin the tube around to stir the bacteria so that it's totally just kind of moving around within all areas of the tube. I then immerse the tube in an ice bath. When I immerse the tube after inoculation, I immerse the tube into an ice bath. Now the agar will solidify very quickly, trapping bacteria in all of the different oxygenic environments within the tube. Now, the bacteria that I put into the molten agar, I can't see it. My agar is still very clear. As the bacteria increase in population, they will start to cause the agar to get really cloudy. Sometimes they'll even start fermenting, producing CO2 gas, and cause the agar to kind of crack open. So where my tube is really cloudy and where my tube is clear, if it's cloudy, I have bacterial growth. If it's clear, I have no bacterial growth. And once I do this with a bunch of different organisms, I can then take a look at where these organisms are growing inside of this aerotolerance tube, and I can determine if it's aerobic, anaerobic, and other classifications. So the classifications that we see for oxygenic requirements include obligate aerobes, and they are obligated to grow in an aerobic environment. So you can see here, they're growing at the top of the agar in this tube. So they're only going to grow where we have a really high concentration of oxygen. My obligate anaerobes are organisms that oxygen becomes toxic to them. So they're only going to grow in the bottom of the tube where there is little to absolutely no oxygen. My obligate anaerobes are difficult to grow in the laboratory because they are, you could say, allergic to oxygen. For them, they get these ROSs, what are called reactive oxygen species, and these are things like superoxides and hydrogen peroxide. Those get created inside of these organisms because they don't have the ability to handle oxygen, and so these molecules here, the superoxides and hydrogen peroxides, are free radicals, and they will bounce around and begin destroying the cells. Now, the other three classifications that we have are facultative, aerotolerant, and microaerophiles. So let's talk about facultative and aerotolerant first. A facultative organism is one that can utilize both aerobic and anaerobic forms of metabolism. Now, aerobic cellular respiration is the most efficient form of ATP production and energy production. So the cells that were trapped in the high oxygen environment are going to use O2. So these guys are using aerobic cellular respiration. They will grow in a much higher concentration. My organisms down at the bottom of the tube have no oxygen or very little, not enough to carry out respiration, so they're going to use fermentation, which is an anaerobic process. So I have both anaerobic and aerobic conditions, both aerobic and anaerobic growth in the same tube. So my facultative anaerobes, because they can use anaerobic, but my facultative microorganisms are able to grow in both. They switch their metabolism. When they get down to the bottom part of the tube, they're like, oh, there's not enough oxygen for cellular respiration, so I'm just going to switch over to fermentation. Aerotolerant organisms will also grow for the most part throughout the tube. They don't usually grow on the surface. There's very little to no growth where oxygen concentrations are really high. But once my oxygen concentration drops at about 3/4 of the way, or about 1/4 of the way down the tube, my bacteria can then begin growing. These are anaerobic organisms that can carry out anaerobic metabolism in the presence of oxygen. They cannot, however, use aerobic cellular respiration, so we won't see them in the very top of the tube. So an anaerobe cannot switch its form of metabolism, an aerotolerant cannot switch its metabolism. It is strictly anaerobic. It can just carry out anaerobic metabolism in the presence of some or low levels of oxygen. So these ROSs, these guys oftentimes produce enzymes that will destroy these free radicals over here, the superoxide and hydrogen peroxide, in order to survive in that low oxygen environment. Now, our last fellow here is the microaerophile, and microaerophiles are the Goldilocks of the aerotolerance world. They are not completely anaerobic, but they're not completely aerobic either. So these would be the ones that fall under neither. Microaerophiles, they need just a small amount of oxygen for growth, but they can't have too much. So they are very, very picky. I call them Goldilocks because one up here is too much oxygen, down here is too little oxygen, in the middle is just right. So these guys require small levels of or low levels of oxygen, but they can't live without the oxygen. So they are neither aerobic or anaerobic, they're somewhere in between. So they are microaerophiles. They need microscopic or micro amounts of oxygen. Aero means oxygen, anaero means no oxygen. So they need very small amounts of oxygen. Other environmental factors of growth include pH and water. Of course, pH, most bacteria like things just slightly on the acidic side, just barely, but just slightly on the acidic side. Neurophiles thrive in a pH range of 5 to 8, so they oftentimes will do well in a slightly acidic environment. Acidophiles like things below 5. Alkalinophiles can thrive in conditions above a pH of 8. Of course, water is a given, right? Water is required for all microorganisms. It contains lots of different soluble minerals and nutrients required. Bacteria do have to maintain some type of osmotic balance with their environment, and that disturbing or taking away that osmotic balance is what's oftentimes done with curing. So meats and cheeses are dried and cured for a long period of time, and that pulls the water out of the environment and helps with preventing growth or inhibiting growth of bacteria in those particular foods. So we can use water. We can use pH too, right? Pickling cucumbers and onions and radishes and all kinds of other vegetables and stuff. Pickling works very well because most pathogenic bacteria and those that cause issues will not survive in a low pH. So when you're pickling, you use vinegar, which usually is a pH well below 5. In our next lecture, we're going to talk about nutritional factors or chemical factors of growth. This is the end of part 2 for chapter 4. I will continue with part 3 and talk about nutritional factors of growth and finish up. Have a great day.